All right. Thank you all, as I just said, for joining us today. As a reminder, the public webinar series is designed to educate attendees on the scientific ecosystem, and each month we'll have a representation from either a data coordinating center, program, or group with new developments from the centers that support them. Today, I'm going to share some KC scientific updates, upcoming events within the ecosystem, and then we'll hand it over to Sreyya from exRNA. If you were unable to attend last month's webinar or any of the previous webinars that we've held over the years, the webinar recording is available on our YouTube page. You can scan the QR code here to bring you to our playlist. If you have any feedback, questions, or you'd like to be added to our listserv, please email us at help@cfdeknowledge.org. So for some upcoming events, I'm going to be talking about the various conferences and different events that we'll be hosting throughout the rest of the fall. And so our major upcoming event is going to be the American Society of Human Genetics, which is going to be October 14th through 18th in Boston. We have a booth, ancillary session, and networking event, which I'll talk to you a little bit more about today. We'd like to formally invite all of you to mix, mingle, and map the Common Fund data ecosystem. This event will be held on the 14th of October from 7 to 9 p.m. in the Westin Boston Seaport District, which is the hotel directly next to the convention center. This networking and engagement event will bring together consortium members, researchers, awardees, and interns for a night of connection, knowledge sharing, and future planning. We'll have lightning talks, poster presentations, demonstrations, and opportunity to learn about funding pathways. This event is available to people who are within the Common Fund ecosystem and without. This is a free event. You do not have to be registered for ASHG to attend. So if you're local in the area and you're not planning on joining for ASHG, please feel free to come stop by for this event regardless. We'd be happy to have as many of you who are from the area nearby. And this is a session that we're going to have to jumpstart ASHG and get us excited for the 3 days of excitement that we have going on at the booth and other talks, which I'll get into in a minute. Registration is live. And I will share the registration link after, but this is basically just to track your name and information. We are not, again, having anyone pay for this. And no, again, registration for ASHG is not required. Again, this is something that we'd like to just track people. And if you would like to post a poster as part of this talk, you can register for a poster as part of the poster session, which will be through a 250-word abstract that you'll submit using this registration here. If you have any questions or concerns about this event, and there's a lot of details that go inside with this, please let me know and I can answer any and all that you may have. And as I mentioned, we're going to be having our booth again this year where we'll be able to see the ecosystem in action. The booth is going to be the 15th through 17th from 9:30 to 4:30 p.m. each day. We're extremely excited about this. We had our first ever conference booth at ASHG last year, and we are going to do it even bigger and better this year. We'll have a spot at booth 1428, and it's going to be your lab for scientific updates, exploration of live tool demonstrations, one-on-one with program leaders, including those from the NIH, and we'll have DCC Spotlight Hour showcasing innovations across the consortium. So don't miss your chance to discover, engage, and network with teams driving the future of data and discovery. We'll also have an ancillary session, which will be held during the ecosystem. And this is going to be October 16th from 11:45 a.m. to 1:15 p.m. in the convention center. We're really excited about this as it's going to be scientific stories, and we will have a little bit more information as this event gets closer. But these are the 3 events that we're extremely excited about, and we're really looking forward to making ASHG a success and have the opportunity to speak and meet with you in person. One other thing that I wanted to highlight is the Decoding the Data System podcast, which is run by the Training Center. And this podcast is more than just a podcast. It's actually a community for anyone who's passionate about the potentials of omics research to solve complex biological puzzles and addressing pressing health challenges. So this is designed for whether you're a seasoned researcher, a student who's just starting out, or you're just curious about biology. So there are 8 episodes that are currently out at the moment. So please tune in to hear more from the different programs in the ecosystem, along with Alyssa Dillman, who is heading this project. So with that, those are the updates that I have for CFDE, and I will now be handing it over to Sreyya from exRNA. So Sreyya, feel free to get started when you're ready. That looks great, Sreyya. Thank you.
Okay, great. I'm assuming everyone can see my screen.
Yes, we can hear and see you great. Thank you.
Great. Okay. Thank you, CFDE, for the opportunity to present here today, and thanks for the introduction. So today I'm going to talk about some of the work that our lab has done on creating a resource for extracellular RNA using RNA sequencing data, and how this resource can be potentially used for future biomarker research. So before I get further, let me explain what extracellular RNAs are. We usually think of RNAs as something confined within cells where it plays a role in protein synthesis and regulation. But it turns out that cells also release RNAs into the extracellular space. And these RNAs are carried inside extracellular vesicles, bound to RNA binding proteins. They are also bound to lipoproteins. And so they remain stable outside cells. And they can be detected in nearly all biofluids, including plasma, CSF, urine, saliva, serum, and so on. So recognizing this potential of exRNAs, the NIH launched the Extracellular RNA Communication Consortium, or ERCC, in 2013, which was an NIH Common Fund program. So the overarching goal of the ERCC is to empower research into exRNA-mediated communication. And this consortium initiated the mapping of extracellular RNA present in human biofluids. And increasing next generation sequencing data sets during that time provided an opportunity to generate this foundational knowledge by creating a common resource for the extracellular RNA data. So the consortium initially produced the first version of the exRNA Atlas in 2019, using about 2,000 RNA-seq profiles that we had in the exRNA Atlas at that time. And in 2023, the Atlas was further extended by a map of extracellular RNA bound to RNA binding proteins. And currently, we have more than 11,000 exRNA profiles in the Atlas. And these profiles are part of 92 data sets spread across 13 human biofluids and includes 25 diseases and conditions. We have a common data processing pipeline optimized for small RNA sequencing data, which makes the RNA sequencing analysis uniform across all these studies. But one problem that we face when analyzing extracellular RNA data is that we don't have extracellular RNA specific annotations. So we have to overlap our sequenced reads with GenCode gene transcripts or other annotations like regulatory region annotations to know more about them or to even give them an identity. And this creates a gap in understanding exRNA biology as a whole. So we set out to create unique identifiers for extracellular RNA loci and basically give it its own identity. And for this, we merged the overlapping extracellular RNA sequence reads from different samples of a biofluid to get these merged loci, which we called as extracellular genomic loci or EXGLs. And we assigned unique identifiers called EXGL IDs to each of these EXGLs. And we did this analysis for 5 biofluids, which were the most represented in our resource. And we identified about 17 million extracellular RNA, extracellular genomic loci in the human genome. To give an example of how this might look like, this is a snippet from the ApoE gene region of the genome in chromosome 19. Here we have laid our plasma EXGL track along with GenCode gene tracks and regulatory element track. And you can see here that now these EXGLs have their own identity, which they didn't previously. And you can see here that these EXGLs actually do not map to any of the GenCode genes or ONCode regulatory elements, and still now we have documented them because they are being transcribed and exported into biofluids. So this example is just for a small region of the genome, and we have this annotation for all the XGLs throughout the genome for the 5 biofluids that we looked at. And this resulted in the creation of an unbiased map of extracellular RNA loci, similar to the likes of the maps of protein coding exons or regulatory regions. And we have submitted a paper on this in July 2025, and this is currently under review. So with the completion of this third and final version of the exRNA Atlas, and the identification of these loci in an unbiased manner across the genome, we wanted to answer a few questions. So firstly, what % of the genome is being transcribed and exported as exRNA in biofluids? So we found that 18% of the genome is being transcribed and exported as exRNA in the 5 biofluids that we looked at. And we also found that a large amount of these, like you can see in the Venn here, about 11% were specific to each biofluid. So about 11% of these EXGLs were specific to each of these biofluids, which you can see in orange here. And less than 1% of this 18% were shared across all biofluids, which you can see, which is represented by the blue bars here. So overall, this is huge that 18% of the genome is being represented in biofluids. And if the genome is so well represented outside cells, can extracellular RNAs serve as potential biomarkers? So for more than a decade now, extracellular RNAs have been linked to a wide variety of diseases. They've been studied in as potential candidate biomarkers in Alzheimer's disease, multiple sclerosis, gliomas, cardiovascular diseases, several cancers, and even in pregnancy complications. So what makes them so appealing is that they can be detected in accessible biofluids like plasma, serum, urine, or even saliva. So this offers a window into tissue states in a minimally invasive manner. And one clear example of the potential of these extracellular RNAs is a clinical test for prostate cancer, the ExoDx EPI test. It's a urine test that looks at the extracellular RNA abundance and uses that to assess whether a patient is at risk for aggressive high-grade prostate cancer. So this test can guide decisions on whether to proceed for prostate biopsy without needing a rectal exam. And this test has now been included in clinical guidelines. And the EPI test provides a more accurate risk assessment for aggressive prostate cancer compared to traditional protein tests like the prostate-specific antigen or the PSA test, and other common existing clinical calculators. And this test is based on transcripts of 3 genes carried in urinary extracellular vesicles. And also, most of the biomarker studies that I showed in the previous slide, especially in cancers and neurodegenerative diseases, being focused on extracellular microRNAs. And this one is focused on the protein coding genes itself. So this raised an important question for us. What is the broader diversity of extracellular RNAs circulating in our biofluids? So if you look at the Venn diagram here, you can see that GenCode gene transcripts map to about 66% of the human genome, and our plasma EXGLs map so these are the exRNA map that we had, the EXGL map. So they map to about 9% of the human genome. And when we overlapped these GenCode transcripts with our EXGL regions, we found that about 6.4% overlapped with the GenCode gene annotations. And when we zoomed into these EXGLs overlapping with the gene annotations, most of them were protein coding RNA and long non-coding RNAs. This is expected also because long non-coding RNAs and protein coding RNAs make up most of the transcript regions in the genome. We included all other RNA biotypes in the rare category here. And we included multiple overlapping annotations for a particular region in the multiple category here. So this rare category included microRNAs, snRNAs, yRNAs, and others. And when we specifically looked at microRNAs in the rare category, we found that they made up just 0.02% of these XGL annotations. So this was particularly surprising because microRNAs are the most researched type of exRNAs, and they comprised of only to 0.02% of the exhales that we observed. And this kind of also tells us that there's possibly so much more potential in looking at all types of exRNA rather than just microRNAs in the biomarker discovery space. And overall, here, about a third of these EXGLs did not lie within gene annotations, within GenCode gene annotations. So we wanted to see if these lie within regulatory element annotations. And a small percentage did overlap with regulatory annotations, but not all. Still about 1/4 of the EXGLs did not overlap with GenCode gene annotations or regulatory region annotations. And this also shows us that these regions are still being transcribed and exported into biofluids, although they haven't been reported in the current gene annotations available to us. So what are these regions? Do they have a functional relevance? Do they represent new biology? At this point, it's an open question for the field, and it highlights why having an extracellular RNA-specific mapping resource is so important. And just to give another side note, we did find some evidence suggestive of positive selection in these EXGL-only regions here. Which we have mentioned in our paper, but I'm not going to go into those details for this talk. So now that we have this huge resource, how may one explore and use the extracellular RNA atlas for their own work? So I would like to introduce the Common Fund, NIH Common Fund Data Ecosystem Knowledge Center here. So the NIH Common Fund Knowledge Center brings together multiple Common Fund programs under one framework, and our exRNA Atlas is also integrated into this Knowledge Center. So the Knowledge Center has developed a user interface for the exRNA data, which connects a user to the extracellular RNA genomic loci, or EXGLs, and all their associated annotations. So first, you can query genes or regions of interest. For example, here I queried ApoE gene linked to Alzheimer's disease. You could alternatively also query your regions of interest. It just doesn't have to be a gene. It could be any loci in the genome. And you have multiple filter options here where you can narrow your search of EXGLs within a particular biofluid, within just a few genes or gene types, regulatory elements, and so on. So this web page is not currently available. It's under development, so it will be publicly available from October to November this year. And once you submit your query, the system returns the extracellular genomic loci, or EXGLs, overlapping these genes across different biofluids. The results show up as both genome browser tracks here, and a table. So here in the genome browser visualization, you can literally see where your EXGL overlap your genes of interest or regions of interest in different biofluids. You also have the table format. You can filter these results on this window, or you can also download them and work with them. And each line here represents an extracellular genomic loci. So once you have these results, you can prioritize EXGLs based on different criteria, like your biofluid of interest, your gene type of interest, and so on. You can also prioritize EXGLs based on exRNA Atlas data sets. So these are the data sets from which we created these EXGLs. So you can tell if an EXGL is present in multiple data sets or just one or two data sets. And you will have hyperlinks from these data sets where you can see the diseases of the samples used in the data sets and other metadata related to these data sets. And if a particular data set is of interest to you, and if there are EXGLs uh, expressed only in that data set, it could be interesting for your work. For example, if an EXGL is present only in an Alzheimer's related data set, it could be a potential candidate biomarker. And another feature you can do is you can sort based on read depth, which is the normalized expression levels of these EXGLs. So you can sort based on this and only look at the EXGLs with the highest expression levels if that is what you are interested in. So this is a broad overview of the features in the CFDE user interface, which will be publicly available in another month or two. We also have other publicly available resources. You can scan the QR code here, which will link to our Atlas Wiki page. And from there, you can explore all of these resources. So you can directly, these are also part of the paper that we are going to publish. So you can directly download EXGL maps with their EXGL IDs and their associated annotations. You also have API access to the EXGL IDs. Um, we have created a UCSC track hub for users. So if someone is familiar with using the UCSC genome browser, they can just load on this track and look at their regions of interest from here. We also have another user interface linked from the exRNA Atlas webpage, which is mentioned in our paper. So these are some of the other resources that you can explore. So that's kind of the end of my talk. So just to summarize the points that I spoke about today, about 18% of the genome is being transcribed and exported as extracellular RNA in 5 biofluids. And microRNAs make up only 0.02% of the EXGLs, and 1/3 of the EXGLs do not lie within gen code annotations. So this opens up the unexplored majority of the extracellular transcriptome for future biomarker discovery. And this atlas as a whole enables the systemic exploration of extracellular RNAs as biomarker candidates that are minimally invasive across biofluids. So with that, I would like to thank my lab, my PI, Alex, and Matt, and my co-author, Rob, and everyone else who's helped with this atlas work, and also the CFDE Knowledge Center team for their huge effort in making this resource possible. And also every one of our collaborators and everyone who's part of the extracellular RNA communication consortium and worked with us for making this resource possible. Thank you very much. Do you have any questions?
Thank you, Sreyya. If anyone has any questions, please feel free to use the question and answer or the chat. You can also raise your hand and I can take you off of mute if you want to ask a question over camera.
Sreyya, thank you. That was fantastic. I guess while we're waiting for some of the questions to trickle in, I wanted to chime in with one of my own. I missed the very beginning of your talk, but I'm curious what is known about these xRNAs during, say, early development versus later in adulthood. Has it been well characterized how stable they are over the course of somebody's life?
Oh, yeah, that's an interesting question. I don't think that's an active area of research yet, but it's definitely something which is interesting to look at. But I'm not quite sure if it's already been studied. As far as I know, I haven't read any studies in that context.
Thank you.
Julie, this is Matt. I'll just add one thing. Yeah, in terms of developmental, I'm not sure much has been looked at. At least I'm not aware of that. There has been a group at NIH has looked at xRNAs through aging. So that has been examined. As far as stability, again, we always have this idea that RNAs are labile and degrade, but a lot of these, because they're packaged either as with either proteins or vesicles, they really can survive fairly well in the circulation. So I think it's going to be a distribution of how those survive. Some may be quick, some may hang around.
Great, thank you.
Any other questions? Like I said, please feel free if you want to raise your hand, use question and answer or the chat. We'll be monitoring all three. Okay, we have one question in the question and answer, which says, great presentation. Have you looked at levels in nasal swabs, another non-invasive specimen?
No, actually, we've only looked at plasma, CSF, serum, saliva, and urine, but we are planning to extend this analysis to pretty much all the biofluids that we have available in the atlas. So we will be looking at other ones, but right now we only have looked at these 5.
Great, thank you. Any other questions?
Guess one more from me, and again, sorry if you covered this at the beginning when before I hopped on. In terms of investigators who are interested in potentially following up on these, are methods sort of well established at this point for studying these xRNAs? Are there other resources that folks could take advantage of that you all have where they could get, protocols and things like that for studying them?
Yeah, so in disease context, in our current paper, we have described a use case for identifying candidate biomarkers in Alzheimer's and Parkinson's, and we have used the resource that we have to kind of identify potential top 1,000 or so biomarkers for uh, Alzheimer's and Parkinson's. So those methods are listed, described in our paper. So if anyone wants to use it for cancers or any other disease, they can just follow the same methods and they'd be able to kind of um, get some idea of potential AXGLs that they should be looking at for their disease of interest, which is kind of like differentially abundant in controls versus their disease.
Yeah, just add to that, Julie, it's a good question. There is a lot of work is done in the ERCC looking at different methods and protocols, library preparation, et cetera. So there is a paper, the ERC published a paper package a few years ago. It's still online at Cell Genomics. So there are some groups that looked in detail, a variety of different protocols. To answer your question, there are also some protocols. Some of those protocols are available on the ERCC portal, so exRNA.org. So to answer your question, so yeah, some people have looked at it. It's evolving too, but there have been a lot of studies looked at different protocols and methods for isolating xRNAs, either from biofluids and different preparations, et cetera. So yeah, that information is out there, and I'm sure it's changing too.
Great, thanks.
Okay, we have Two other questions. Oh, Shreya, did you want to add to that? Sorry.
Yeah, I just wanted to add that for the protocols and the wet lab part, our lab is also working on another paper where we are using this resource to look at like differences in expression levels between different kits, like different library kits, RNA extraction kits. We're comparing those. So yeah, we're working on like trying to see maybe provide a resource for people to prioritize which kits to use for their disease of interest.
Great. Doug Keel has a question in the Q&A, which is, what is the variability of XGLs across samples?
Um, so I don't have an exact number on that, but I must say there is quite some amount of variability even between individual samples. Um, but having said that, there's still within a particular data set. So if a particular data set is from individuals with Alzheimer's of a certain age, and compared to another data set of cancer or something, you do see um, some amount of commonality in the Alzheimer's data set versus the cancer data set, but I don't have a number put on inter-individual variability, but there is some, especially because we are looking at millions of these regions across the genome. So there is going to be a lot of variability even between individuals.
Great, thank you. And then there's one other question from an anonymous attendee. What do you think xRNAs are doing? What is their mechanism of effect?
So okay, so that area is being heavily researched right now. So the idea is that they are released out of cells. So if an RNA is present in a cell and it is released out of cells and it is like moving into another cell and affecting the regulation of the other cell, it's being studied in cellular communication in general. So there have been many studies that have shown that RNAs play a role. Extracellular RNAs that move out of their cell of origin play a role in intercellular communication and regulating the expression levels of other cells.
Great. Thank you. If there are any other questions, like I said, please feel free to use question and answer or raise your hand. We definitely have some time for some more if there are any other questions. Okay, it looks like that is all for today. So thank you so much to Shreya and xRNA for joining us today for our webinar series. This is just a reminder to everyone, our next webinar will be September 26th, and we hope to see you then. So thank you so much again, Shreya. We really appreciate it.
Thank you.
Thanks, everyone. Bye.